Okay, everyone, so I think we'll go ahead and get started on the seminar for today. We're going to start off, as usual, by a reading of the land acknowledgement. So we respectfully acknowledge the territory in which we gather as the ancestral homelands of the Beothuk and the island of Newfoundland as the ancestral homelands of the Mi'kmaq and the Beothuk. We would also like to recognize the Inuit of Nunatsiavut and Nunatukavut and the Inuit of Nasinan and their ancestors as the original people of Labrador. We strive for respectful relationships with all the peoples of this province as we search for collective healing and true reconciliation and honor this beautiful land together. So today for our seminar, we would like to welcome Dr. Daria Gallardi from DFO, and she's going to be talking to us today about blue mussels. So welcome, Dr. Gallardi, and feel free to start.
Thank you. I hope so. Okay. So today, again, my name is Daria Gallardi, and today we are going to look into putting some mussel into it and the DFO work on the environmental condition on Newfoundland cultured blue mussels. So the first part of my presentation is going to be treated a little bit like a recipe. So we'll see what are the ingredients that we really need to do some mussel work and some mussel research. So first of all, you really need a team of dedicated people that really want to do some research. And as you probably read, I work at Fisheries and Oceans Canada, and a lot of the work that we are doing there on mussel is funded by the Aquaculture Collaborative Research and Development Program. In DFO, I work with Dr. Harry Murray in particular, but we also collaborate with Memorial University, and some of the work that I present you here, it was my PhD work, and my supervisor at the time was Dr. Chris Parrish. And also recently we did some collaboration with Dr. Matt Rice and Zizou in his laboratory. We also had always a industry partner that we worked with, Norlanti Processor and Thimble Bay Mussel Farm, because we need a place where we go sampling and we need a place to give us the mussels. So they always work with us very kindly. And finally, during my PhD, I also had some extra money coming in that came from the Research and Development Corporation of Newfoundland, and really helped in my work. And after this, what's the next ingredient that we need? Of course, the mussels. So why are we interested in looking into mussel aquaculture research? So mussel and bivalve more in general have a really close relationship with the environment. Is a two-way relationship because bivalve are considered a keystone species in the ecosystem because they exert a top-down and a bottom-up control on the environment. So you can imagine that while we concentrate a lot of bivalve in a specific location for aquaculture, we can have large effect on the environment that can go from effect on the nutrients in the water column, we can have deposition and effect on the benthic habitat. We can possibly introduce pest and disease with the movement of the bivalve. And you can also have effect on other marine species like marine mammals that can get entangled or can be excluded by certain location. And birds, marine birds can be attracted by the farm sometimes, for example. So, however, when working with the bivalve aquaculture, there is also something that tweak the story a little bit, because bivalve can be also used to get some positive effect on the environment, such as the counteraction of eutrophication, so the excess of nitrogen in the water, for example. And bivalve aquaculture has been used in different places, coupling with the restoration or remediation projects too. So there is this difference with finfish aquaculture that we can actually add an environmental value to bivalve and in our case, mussel aquaculture. So the next ingredient that we need is a place to do our work. So as I said, I work for Fisheries and Oceans, and right now the work that we do on mussel aquaculture is done under two main boxes. One is the Aquaculture Monitoring and Modeling Program, or AMMP. Which is monitoring the impact of both finfish and shellfish aquaculture, and is looking in particularly a far field effect of the aquaculture farm on the environment. And as I mentioned before, the other big box is the Aquaculture Collaborative Research and Development Program, or ACRDP. You must know that at DFO, they really love acronyms, so everything as an acronym, they never call the things with their full name. So the ACRDP project are projects that have a mandatory collaboration with the industry, and they can go from 1 to 3 year maximum of length. Finally, we also need a question or some question that we want to ask is our last ingredient. So what I'm really interested in too is what is the relationship between the environment and the cultured blue mussel. And in our case, we work with Mytilus aedulis in between the blue mussels. So I go back to that two-way arrow at the relationship with the environment. So what is the aquaculture input in the environment? And for that, we discussed that under the AMMP. And also what in exchange, how in exchange the environment is affecting the mussel. And knowing that, how we can utilize the environments to obtain a productive and sustainable mussel aquaculture in Newfoundland. Productive means that we need to get a good product that is lucrative for the farming, but also we want a product that is integrated with the environment and then is giving the least possible effect on the environment. And about that, we will discuss all the work that we did on culture depth of cultured mussels. So very briefly, the Aquaculture Monitoring and Modeling Program is a nationwide DFO initiative that was started in 2017. As I said, it's both in finfish and shellfish. And the main objective of this work were to develop and implement a monitoring program. And once we collect data during the monitoring program, those data are going to go to feed models, and those models are going to be very useful to predict future trends, in particularly under changing climate condition. And the final goal of this program is to provide a scientific advice to the management in DFO on the long-term sustainability of aquaculture. What do we do for this program? I show you here just an example. This is like the site that we sample for our mussel part of the program. And all the little dots that you see here are stations that get sampled. And the pink part is the aquaculture leads. So that's the part where the mussel are actually located in the bay. So you see that all the stations that we sample are inside, in between the mussels, but they're also progressively outside and further from the farm to see the difference in the effect of the farm. In all these stations, we measure a certain number of parameters. So we do work on physical oceanography. So in the bay, there are DCPs that they measure currents and they're left there for a year generally. And then at each station, we do CTD profiles that measure temperature and salinity. And recently also, we have also fluorometers to measure also chlorophyll A. Then we take water sample with Niskin bottle and with zooplankton nets. And on those samples, we do different analyses. We look at total suspended solids, chlorophyll content, carbon and nitrogen content. And then we look at phytoplankton and zooplankton taxonomy, and we're attempting also zooplankton biomass. At the beginning of this project, we did also some benthic sampling, but right now has been excluded for the bivalve work because it seems not being particularly interesting, but is actually a fundamental part in the finfish part of the program. So for this program, there is no results really because this is an ongoing thing, and the results will be at the end of many years of experimental work to figure out a national scale monitoring program can be applied for the years to come to aquaculture in order to give to Canadians an hotspot of aquaculture impacts on the environment in all the country. So the longest part of my talk is going to be now on the work that we did on culture depth. So we'll present you here two main research that we did. One was long, 2 years, and was done between 2012 and 2014. And then we had another year in the same location with the same experimental setup that was in 2016. So you see here in the bay is the same one that I showed you in the previous map. But there are three blue dots. Those three blue dots were what we call the shallow water site. That meant that the mussel, they are suspended in the water column. So the headline for the mussel line, it was a 5 meter. And then there is a sock that is long 5 meter. So the mussel in this case were suspended between 5 and 10 meter in the water column. And then we have other three sites, the red dots, that were what we call the deep water sites, where the headline was a 15 meter, so it was deeper, and the mussel went from 15 to 20 meter in the water column. Why are we interested in looking at culture depth? Well, this is because most of the mussel aquaculture here in Canada is suspended, and it is done at a depth around 5 meters, sometimes even 2 meters. But a lot of other locations in Canada have a much shallower site. We have a deeper site compared, for example, to PEI. So we have the possibility to go deeper in the water column for a number of reasons. First, it seems to be a more stable environment for the mussel to grow because it's less affected by the land runoff. So, for example, the excess of nitrogen, the rain, fresh water coming in from rivers. It can be also less affected by weather conditions, such like during storms, high wind and high waves. So the idea is that the mussel have a quieter, nicer environment all year round, and they can grow better in it. But also we are trying to move away from the close coastal locations. That is the busiest one, and there is a lot of other users in the coastal water. Plus, there is this idea that in deeper sites and in deeper water, there is a slightly higher current that can disperse better the deposition of the farm. So we're not going to create an anoxic layer just right under the farm, because the current is going to help to bring away whatever is produced by the mussels. So in this particular site where we're looking at, and here I point out this one of the site that we look at here on the top that is a little further away from the other one. For that particular site, there was also another reason why the farmer wanted to go to deeper water. This is because in the spring, there is packed ice coming into that location. And therefore, when you have your headline of 5 meters, there is a really good chance that the packed ice come in and it is gonna drag all your lines, you're gonna lose all your mussel. While if you put them deeper, there is way less chance that there's gonna be a huge, enormous, big piece of packed ice that is gonna take your line away. So during those experiments in these 6 sites, well, we measure different kind of parameters. So we looked at the environment again, and we look at temperature and chlorophyll A, and then other things like currents, for example, and salinity. Then we look at the mussel, and in the mussel, we looked at their condition and weight, and then we look at their reproductive organs. And we also look at their biochemistry, so their content of lipid, fatty acid, and glycogen. And finally, we did some transcriptomics work on them. So I'm going to present you here just the snapshot of the main result that we get from all these different parameters. So for the environment, on the top graph, we have the temperature, and on the bottom graph, the chlorophyll A. I'm sorry about the fact that the left and the right graph have a different scale, but they come from 2 different articles, so they were not done at the same time. And unfortunately, also the symbol from deep and shallow water is actually reversed in the 2 graphs, but bear with me, I'm going to point out what we need to look at. On the top, you see also the different year of the experiment. So what we want to take home from this graph of the temperature on the top is that every year there is a pretty big part of the year from May to September, October, where the mussel in shallow water have warmer water compared to deep water. That period is the period where the water is stratified and we have the formation of the thermocline. So we see that the shallow water mussel are in a different layer compared to the deep water. And this difference sometimes is actually pretty high. It can go up to 5 degrees here in the average, but if you go and look at the data actually when we measured without the monthly mean, there are differences at some point in time of 10 degrees in temperature. For the rest of the year, like late fall and winter, where the water is mixed, you see that shallow and deep water sites have the same temperature. For the chlorophyll A on the bottom, chlorophyll A is a proxy that we use for phytoplankton. It's a proxy, it's not perfect, but we can still take some message away from it. So we don't see differences between shallow and deep water for the chlorophyll A because we have a lot of standard deviation, so there is no real a significant difference between the two kind of sites. However, we do see a very clear seasonality. Those peaks that they are highlighted there are the spring bloom that it generally starts around February and go up to May more or less. And this is true for 2 of the 3 years that we look at. But if you point out this specific year, that's 2014, you will see that the bloom is much delayed. It's actually starting towards the end of March, April, and then it's only going to go up to May. And it has a peak that is less wide and is also shorter than the other years. If you were here in 2014, you probably remember that that was defined as the winter from hell here in Newfoundland. So it was an unusually cold winter. A lot of frozen water around. The bay where we work got completely frozen from January to April, and we were not able to sample in the winter due to that. And that generally doesn't happen in that bay. It's a very rare occurrence. So we start to see that weather actually impacted the phytoplankton bloom, and then we will see later on how this impacted the mussels. When we looked at the mussel, the first thing that we measure all the time is the condition. So, based on the weight. And what we're seeing here on the left, there is a graph of the first experiment that was 2 years long that we did. And then the second one is on the right. During the first experiment that we did, we measured the mussel every 4 months. And in this case, we see that there is no difference between shallow and deep water in the condition. However, we do see a big, clear seasonality trend. So here, this sampling, it was done in May, and that is after the spring bloom and right before the spawning of the mussel. So we see that the condition is at the highest point, and then you see in September, 4 months later, that is after the spawning, we'll see that the condition is much lower, drops down. However, in the following experiment, we figured that maybe we're missing a lot of things in between there because we are only sampling every 4 months. So the second time, we did sample for 1 year every month, and what we see here is that there are actually differences between shallow and deep water. So you see that at the start of sampling in May, although the deep water has slightly lower condition index compared to the shallow water, then in the 2 following months, June and July, we'll see that actually deep water has a better condition compared to shallow water. And then in August, they both have a lower condition after the spawning, and they are getting both shallow and deep water, they are very similar. So remember this graph because we're gonna come back to this later on. So the take-home points from these two graphs are that the condition varies between season and depth, and also something that I didn't show you with the graph, but it was very interesting to find out, is that we found out that shallow water mussel had longer and heavier shells, and also female had a longer and heavier shell than male. And it was quite curious. And the last things to pick up from this graph is that there are differences between years. So you see that 2014 years, we noticed that the condition index, even in May, the nicest months just before the spawning after the spring bloom, is still has a little lower condition compared to the previous year. So we already start to see the repercussion of that very particular winter on the mussels. So what we did after, we looked at the biochemistry of the mussel, and I'm going to present you here just the fatty acid results, because the fatty acid profile is really interesting and go really well with the environment, and it gives a lot of really interesting information, I think. So this is a principal coordinate analysis, and what we are doing here, we are trying to look at how the sample group in this graph based on their fatty acid profile. So the first thing that we see from this graph is that the fatty acid, based on the fatty acid profile, the sample group by season. So this doesn't seem to be an effect with depth, but it's just a season. So what is driving the location or all the sample in this graph are the essential fatty acid, not surprisingly. So we'll see arachidonic acid, and we see in particular the docosahexaenoic acid, or DHA, and the eicosapentaenoic acid, or EPA. So we notice that in the fall, there is the sample locate, those two are both in the fall, September. You see they locate more closely to the DHA, while towards the spring, they are very skewed toward the EPA. So what is happening here, it's easily explained because that peak that we saw in the chlorophyll A, that's the spring bloom, is very rich of diatoms, which are the main source for mussel of EPA. So that's why in the spring we have this mussel rich of EPA, while the smaller fall bloom is rich in dinoflagellate, which are the main source for the mussel for DHA. So that's why the fall sample located more towards the DHA. The other thing to point out here really briefly is this group of deep water mussels that they kind of like move away from the other sample and is a false sampling, but they're a little skewed towards the APA, although they are taken in the fall. Those mussels were the one site that I point you out that it was further away in the bay, and that sometimes acts really interestingly compared to the other sites that we were looking at. So this was the first year of the experiment. Then we look at the second year. The second year, we don't have the winter sampling because we couldn't sample due to the ice. So we have the two fall sampling and initial and final, and then we had the spring sampling in the middle. We see again, same importance of the essential fatty acid in grouping our samples, but we also see some other fatty acid that were not present in the first year. All these fatty acid here are bacterial fatty acid. It's very interesting because bacterial fatty acid are only 4 or 5% of the fatty acid profile of the mussel. However, in the second year, even being a small part of their profile, they were important and were driving the differences between season. So we're going to come back to this in a second. The last things I want to point to you here is this group again of deep water mussel, bigger than in the first year, that is going towards the APA and not towards the DHA like the other shallow water sample do in the fall. And in this case, during this year, it's all the deep water mussel that were actually skewed toward the right side of the graph. So when we look at each single fatty acid or group of fatty acid in the analysis, we found out that the deep water mussel during this year had higher EPA, and they also therefore had a higher content of omega-3 compared to shallow water. However, shallow water mussel had higher bacterial fatty acids. So the bacterial fatty acids are very interesting here because what I think it happened in this situation is that that very weird winter, the shorter spring bloom, less feeding, something happened that affected the shallow water more than deep water during that year. So the deep water mussel had a really nice feeding. They had all their omega-3s, they were pretty happy. In shallow water, I think they were missing something because generally mussel, they don't love too much bacteria. They're too small and they don't tend to be very nutritious for them. So they generally filter them and expel them with the pseudofeces. They don't digest them. However, it has been shown that if they really need some nutrition, if they really need some food, then they are able to intake and digest the bacterial fatty acid too. So that's what I think it happened in this situation. The shallow water mussel were missing something. So they intake more bacteria, while the deep water, they didn't need that because they were already doing great. All right. So after that, we looked also at the reproduction of the mussels. So the reproductive organ in the mussel is the mantle. The mantle is that thin layer that is inside attached to the shell when you open a mussel. When the mussels are spawning, the mantle gets thicker and a little chunkier. However, when the mussels are not spawning, it's a very, very thin layer, almost translucent, and it's really hard to get and it's to sample because it breaks up really easily. So the mantle is not only the reproductive organ in mussel, it has a lot of other function, but we are going to concentrate on the reproductive function. So I don't know if you know much about mussel, but mussel are interesting because male and female can be recognized quite easily during the spawning, because the female is orange and the male is more pale. Now the picture here is a very extreme case. It's not like that all the time, but during the spawning it's fairly easy to recognize them. However, after when they start finishing spawning and when they are spent, they both start to get like beigey or yellowish, so you can differentiate them very well at all. So for this part of the work that we did, we had to go and look at each mussel with histology to be sure if they were male or female, because we needed to separate them by sex. So here you see, this is where the mussel are spawning a female, really nice eggs, pink in color, very cute. And here the male with the little small dots, the sperm that are purple. And when we go towards the spent time, this is probably not yet spent, but it is towards the end of it, of the spawning, we see a lot of more connected tissue, empty spaces where the eggs were, and there is actually nothing when you look at your slide. For the male, we see maybe some new sperm forming, some dots, but you don't see the fullness that you were seeing when the mussel were in full spawning or just before the spawning. So what we did for the reproduction, we looked at the mantle dry weight, because the mantle again is the reproductive organ. So that's what we look at. And we also looked at the gonadosomatics index, which is the ratio between the mantle dry weight and the body weight, the total body weight of the mussel dry. Probably this graph reminds you something, the condition graph that I showed you in one of the first slide about the culture depth. So what we are seeing here is that there are differences between depths, and there are differences in spawning length and spawning timing. So if you see, the shallow water mussel starts spawning right away in May, because there you can see the next months that their condition and their mantle weight went down. So it means that they are spawning. And they go through it a little bit trickling until August. While the deep water mussel, they keep maintaining their condition, it was on the other slide, and they maintain their mantle weight until just the last month, July. And then in August, they go both back to the same condition and to the same mantle weight and GSI. So this may seem nothing, but from the point of view of a farmer, this is a huge point, because if you're selling your mussel in the summer, you better sell your deep water mussel, because they maintain their condition much longer and the spawning is much shorter than in the shallow water. So you are selling a product that is heavier, and that is also much nicer for the people that eat them, because it's much plumper and it's nicer mussel. So good point for them. What we did next is we tried to isolate those mussels that were spawning. So when both shallow and deep water were spawning, so we isolated the mussel in July. Well, the shallow water was towards the end of the spawning, and that was the month where the deep water was all spawning. So what we did was we sent sample away to do an RNA sequencing. Then the results from the sequencing were looked at for a very long time, and we tried to isolate transcripts that they were significantly upregulated or downregulated in deep water compared to shallow water. And of all those transcripts, then we picked 13 genes that we wanted to validate with a qPCR experiment. So what we found out was somewhat interesting, because this is another principal coordinate analysis here. And again, what we are seeing here, we are doing the same thing that we were looking for in the other graph, how the sample group based on their gene expression profile of the 13 genes that we looked at. So what we see here is that they group by sex. So there is no effect of depth in this graph. And the other interesting point to take home from this graph is that all the male are much tightly grouped than the female, that they have a much larger area. So male mussel during this spawning event seems to be much more similar between each other for gene expression profile compared to the females. What is the the gene that carried the most importance in separating, the most weight in separating the sample in this graph. This gene is the KLHL10 or Kelch-like protein 10. That gene is the principal coordinate one. So that gene alone is explaining actually 70% of the total variation between samples. So it's a pretty important thing. Kelch-like protein is a key marker of male gametogenesis, but it has been shown especially in mammals. And it has been identified in other bivalve species before. However, until now, nobody has really tried to quantify it, and nobody has really looked at it in separate sexes, like in this case we did, and in Mytilus in particular. So our results may mean that Kelch-like protein has a role in sperm production in Mytilus like it has been shown in other species before. Need some more work on it, but that is a good start. Another very important gene in this analysis was the NTCO1 or cytochrome C oxidase subunit 1. This gene is involved in different processes and like cellular respiration and oxidative stress, but also in sperm fitness. It's been shown that he helps the integrity of the sperm. Again, it's not in Mytilus before, but in other species. So what we're thinking here that probably, again, a similar function for mussel in this case, and possibly because male mussel have external fertilization. So the sperm has to be fairly resistant in the environment before the fertilization happens. So maybe this gene is involved in that resistance or to external stressor for the male sperm. The other interesting thing here that not only this MTCO1 was important to separate male and female, but it was also one of the two only genes that resulted significant for depth too. So deep water mussel had upregulated MTCO1 compared to shallow water mussel. And this, we think, may be due to the fact that MTCO1 is involved in the response to oxidative stress. So possibly the deep water mussel, if you remember, for a certain part of the year, they live in a colder water, and the oxygen saturation in colder water is higher. So maybe that's the reason why deep water mussel may need an upregulation of this gene that is help in counteracting the oxidative stress. So finally, the last things that we did for this experiment was to look at the shell, because if you remember at the beginning, I told you that we found out that there were shallow water mussel and female had longer and heavier shell. So we did a little side project to measure also the thickness of the shell to see if we could find a difference in there too. And we used an indirect and one direct method to measure the thickness. So the indirect method is the shell thickness index, or XTI, and then the formula there is what is used to calculate it. And for the direct measurement, we developed a little protocol to measure the shell thickness on a scanning electron microscope, so really very precisely. So we develop a protocol to cut always in the same location of the shell, a 5x5 millimeter square that was mounted, and then we measure the thickness. So this little side project took a life of its own. We found actually out that when we were measuring the thickness with the STI, we had significant differences between depth. So we were finding that shallow water shells were thicker than deep water shell. However, with the direct, very precise measurement of the shell, there was no significant difference in depth. So at the end, we didn't really explain much of why our female or our shallow water mussel have a heavier, thicker shell, but this whole thing became a cautionary tale about using an index because we found out that our population of mussel had a weaker relationship between the direct measurement of the thickness and the STI that it was shown in literature, and also our population had a very weaker relationship between the measured length and the STI compared to what was shown in literature. So if you remember the formula that I showed you before, the length is carrying a lot of weight in that formula. So if that is not going to fit there, the whole formula is not going to fit. And also, we did found that male and female mussel have a different shell for different weight for the shell. So maybe that needs to be taken in consideration when you build a formula for an indirect method for thickness. So the whole idea of here is like, well, you really need to develop your own index for the population that you are studying, or at least you really have to quality control very carefully an index before you use it and you apply to your population when you don't know if it's gonna fit. Well, and the last things that we want to take out from this little side project is the fact that maybe when we're going to look at the shell thickness and in study where they're looking at the climate changing condition and in particular increased acidification, then yeah, we want to be really precise with the measurement because we are talking about very tiny changes. So maybe using the scanning electron microscope to measure your thickness is not a bad idea. And although the scanning electron microscope has been used with other bivalve before to measure thickness, as far as we know, we were the first one to try to measure that in mussel, actually. And you may say, well, not everybody has a scanning electron microscope, and yes, that's true. However, a lot of the places where there's some work on acidification and the effect on bivalve shell is done, they do have a scanning electron microscope because they already look at the deposition of calcium carbonate on the on the shell matrix with the scanning electron microscope, so they could potentially apply this method to measure the thickness. So to give a general conclusion on what we found out in these years of experiment on the deep water, on the culture depth, we found out that there are differences between years, between season, and sometimes between depths. So the difference among years is mainly driven by the environment. The difference in season is driven a lot of time by the reproductive cycle and by the environment. And also, those two, the environment and the reproductive cycle together, may like the possibility to use deep water here in Newfoundland because because at the worst, your deep water mussel seems to be similar to the shallow water one, but in certain situations, like we saw that very weird winter in here, we saw that the deep water mussel were actually doing better for their fatty acid profile there, and they were keeping their weight longer during the spawning. So deep water can be beneficial in certain situations. However, there are still a lot of questions that remain, and that would be nice to answer one day. One of them is the quality of food that is bugging me since we did the fatty acid analysis. So the chlorophyll A, if you remember the first graph, didn't show any differences between shallow and deep water. However, we see differences in the fatty acid profile. So that means that probably it's not the quantity of food that is a problem, is the quality of the food and maybe the size of the food could be. So it would be really nice to look precisely into what we have there and to find where the difference is located. Then with that, we looked at the reproduction shift between shallow and deep water. Why are the mussel in deep water spawning later and have a shorter spawning? We may explain that partially with the fact that they are in a colder temperature, but I don't think it's the only reason behind that. So it would be really nice to look into that a little bit more. And then again, these things about the shell, I really would like to know why the shell are different, but again, maybe one day we'll be able to figure that out. And by the way, those are diatoms that are on the top of a mussel shell seen on the scanning electron microscope. So where are we going from here? What are we doing next? Well, finally, it looks like we are going to look into the food quality next, because we are starting a collaboration with Dr. Cynthia McKenzie at DFO, which has a flow cam, and she has the experience to look at phytoplankton identification. So we're going to try to see the different species. First, we're going to have samples that are taken in different months to see the seasonality, and hopefully we'll be able maybe to get different depth sample to see if that is explaining our fatty acid differences. Then the RNA sequencing results, they were exploited up to a certain point. We still have a lot of data that can be taken out from those results, because we only looked at transcripts that they were significantly up or down regulated in deep water compared to shallow water. However, there is a whole other series of transcripts that they are up or down regulated in female compared to males that we didn't look at yet. And also we concentrated a lot on reproductive, on the reproduction side. So a lot of the genes were involved in strictly involved in reproduction, but it would be also nice to look more at genes that they are involved in environmental response in the mussel. And finally, near and dear to my heart is something that is many years that I want to work in, and that's the ecological carrying capacity of a blue mussel in Newfoundland. So the ecological carrying capacity is the density of a cultured organism above which unacceptable environmental and ecosystem effects appear. So work on carrying capacity have been done in other regions of Canada. However, here in Newfoundland, nothing has been done yet on this topic, and maybe we will succeed at some point to work on this. So, whoop. You can change page. Uh-oh. Oh, here. So this is all nice and well, but we need to get something out. It'll be fun to work with this, but we need to get something out of our experiment. We have to, we work for the government, so we have to give something to our stakeholders. That means all the Canadians. So really, what is the relevance of what we did? We did find out some new knowledge for mussel aquaculture. In particularly the fact that deep water is possible and sometimes beneficial here in Newfoundland. We also did the first comprehensive study of the environmental condition in South Arm, New Bay, Notre Dame Bay. And those environmental conditions that we look at are, yes, applicable to aquaculture, but they also can be used by any other users of that body of water. So it's a wealth of information, and it's a wealth of information here of sampling that we can also create an environmental baseline that can be applied for work like the one on carrying capacity that we would like to go into in the future. So I can go back full circle to one of the first slides that I show you, at that tight two-sided relationship between the environment and bivalve aquaculture. So our work might be one tiny little piece of a million pieces puzzle, but it is one tiny little piece to add to that, to put in position in that puzzle that is the relationship of the aquaculture with the environment, and the impact of aquaculture on the environment, and the impact on the environment on the mussels. So this is the reference that I use, and I really would like, before handing, to thanks a lot of people, because of course I didn't do this alone, not at all. So first of all, I would like to thank my supervisors and co-authors, Dr. Harry Murray, Dr. Chris Parrish, Dr. Matt Rice, Terry Mills, which is the owner of the farm where we worked, and Sebastian Donnet at DFO, and Z2 at the Ocean Science. And then I would like to thank Genome Quebec for the work on the RNA sequencing, and Orlantic Processor and Timber Bay Blue Mussels farm employee that put up with us all the time they went sampling. They helped us, and they were always so kind. And finally, acknowledging the funding sources, the LCRDP program and the Research and Development Corporation of Newfoundland. And that is it.
Thank you. That was really great. Thank you for that. I have a question, but does anybody else want to bring any questions forward? I can start with mine, and if somebody else wants to speak up, they can feel free to. So you mentioned going forward that you're going to be looking at the food quality and phytoplankton, and that you're going to be using the flow cam. I was just wondering why you decided to use the flow cam versus doing any measurements for, like phosphorus or nitrogen content or something like that of the phytoplankton.
That's an interesting question. We did do we took some water sample and we actually to give to someone else to do some nutrients analysis before. However, I think that, we didn't add a lot of sample and not a lot of replicates. So the results that we got were not really telling much. And we also, I think we were sampling at the time every 4 months. So we didn't really had a lot of, we needed more sample. And as of right now, we have the opportunity through the aquaculture monitoring and modeling program to get more water sample because we go out for that program. And through that, we figured that one of the things that we can do on the side, because that's not required by the program, is to take some sample for us and looked at that. And we have we have like this, like Dr. Cynthia McKenzie in DFO that is really like a plankton person. So she's really good at it and she's gonna be able to train us for that. So I think it's just like a convenience, to look into that. And I guess that going back to the nutrient work would be interesting too. But as of right now, I don't know if because it's not really something that is funded, is a side things that we're doing. So to run the nutrient, I think it will have to have some more money to add to the thing, right? Because we give them to the group, the oceanography section in DFO, and they have to use their machines, they have to use their people. So we need to give them something. They can't do it for free all the time, especially if it's a lot of sample. But definitely, if we are going to go into if we manage to succeed to do some carrying capacity work, well, definitely something that we're going to look into nutrients. We're going to look into that too. But we need funding, so we're waiting for that.
Okay, thank you.
You're welcome.
So Daria, I have a question. Well, first of all, nice presentation. I'm glad that you're sticking in this area and helping to push back the frontiers. Just I guess a comment and then followed by a more general question. And the comment was your PCOs are fatty acids. You're absolutely right. They're very clear what's going on there. And you, as you know, you pointed out the essential fatty acids, and so EPA and DHA, the omega-3s came up. But interestingly, so did ARA, right? The omega-6 fatty acid was quite important. And is that possibly coming from macroalgae? Is that the supposed source?
I don't know. I never looked into it, actually. It's a good question. It's something I have to look into. That could be an interesting question, yeah. And honestly, I just left the arachidonic acid on this side because, in aquaculture, there is this thing that we always looked at DHA and EPA for some reason.
Yeah.
But, I need to check that. That's a good question.
So arachidonic acid is never present in much quantity, but as you point out, it is an essential fatty acid. And so even though you don't need much, you do need some. So that might be something to think about. Maybe, with all the ice and the wave action, macroalgae breaking up, and maybe that's how it gets in. I don't know.
It is a possibility because there are some of the lines, not all of them, but some of the lines of the mussel, they do have some algae on them when we pull them up. So there are algae around.
Oh, there you go. There you got your source. And I guess the other sort of general question I had was, so IMTA, integrated multi-trophic aquaculture, you just kind of indicated at the beginning of your talk that there's sort of this environmental side that mussels can be put to. Is DFO doing anything with IMTA now? They started off in New Brunswick. Has it ever gone anywhere?
I think that in New Brunswick they're still maybe doing something, but we in Newfoundland nobody ever done any work on it. And actually, it's interesting that you asked because last month some of our technicians came back from the south coast to go to retrieve some oceanographic instruments, and they were in 3 different finfish sites. And in one of them only, the instrument was full of mussel.
Oh, interesting.
I was like, yeah, next time, bring the mussel back.
Yeah.
Because I'm sure they're gonna eat some fish food in there.
Absolutely. Yeah,
So it's very, very interesting because you could see the difference in the three. There were actually 3 sites, and one had full mussel, the other one just a little bit, and the other one had algae. So
right, that would be the total IMTA then, wouldn't it? You'd have your mussels and your algae.
right.
Perfect.
It would have been an interesting side project to look at, but unfortunately they couldn't take the mussel at that time. We need to have a permit to bring them back because it's a finfish site and everything. So, but we'll see. Maybe in the future we can figure something out about that.
Yeah, that'd be cool. There's something that came up in the chat for you.
Nope.
Yeah, Ignacio's having some trouble with the microphone on the computer in the Challenger room. So I think he put some questions in the chat.
Okay, how am I gonna look at the chat? Can I end the screen sharing maybe?
I can read it for you.
Okay, yeah, sure.
It says, in terms of markers that you were using, did you find or look for any related to shell thickness? And then probably also could be linked to X as you show for gametogenesis, or that difference could be more related to environmental conditions. So I guess if you looked for any markers for the shell thickness.
We did have at the beginning one marker, one gene that we were thinking to look at that is perlucin, that is actually related to the shell, and that would have been really cool to look at. However, we didn't manage to have a primer set that worked for our qPCR. So in the RNA sequencing, we did add something, and that was the only one that came up in the analysis that was done until now. But that is, I didn't mention that, but that is a really good question because we, I think that in between the environmental, the gene that they are more related to environmental response, also the gene that they are related to shell formation will be really good to look at. You know, even just for an interest in acidification, possibly, like it would be really nice to exploit those RNA sequencing data to see if we can find more genes that they are involved in the shell for sure.
Does that answer your question? Yeah. Are there any other questions?
Sorry.
You can put it in the chat if you don't want to. Oh, not him.
Very nice talk, Daria. Thank you.
You're welcome.
In slide 12, you have the PCOA and then the table that shows the higher bacterial fatty acids in the mussels compared with deep.
Yeah.
Did you look at the concentration of bacteria in the shallow water compared with deep water?
No.
Would it be fair to say that the shallow water mussels are exposed to higher concentrations of bacteria than deep?
No, unfortunately not, because that is a thing that came out at the after, like when we looked at the data, and that was the second year after all the project was done and all the sampling was done. So for the main scope, I guess, of the project at the beginning, we didn't really put any of that on the, and need to look at bacteria in the water column. However, again, that is something that it can be looked into once we move towards some work on carrying capacity, for example. That is would be something that can be included in there. But for that particular project, if we would have known maybe after the first year a difference like that, maybe we could have added in the second year of the experiment. But unfortunately, we saw that when everything was all done.
Right, but if the shallow mussels are in warmer water, bacteria or higher concentration of bacteria, could that just be why you see a higher bacterial fatty acid signature in the tissues, just because they're in much higher abundance or concentration of bacteria in their environment because of the warm water?
Yeah, it's possible. And again, the explanation that I give is just my general explanation for the fact that, you know, I mean, we are also talking about a percentage of fatty acid here. We have to remember well. And so when you see like the omega-3s goes down and the bacterial fatty acid goes up, it's because the whole profile is 100%. So you have to consider that too. But yeah, it could be, again, a lot, like, I don't think that the water temperature explains everything we see between shallow and deep water. but it could explain part of the differences for sure, especially with the reproduction, because that's reproduction is both food and temperature related in bivalve, right? So
Oh, that's good. Thank you.
you're welcome.
I would just add to that if you really wanted to get at it, you could do LCMS and figure out where the bacterial fatty acids are. And if they're associated with triglycerides, then it has to be inside the animal because bacteria don't make triglycerides. If they're associated with phospholipids, then it's just basically intact bacteria that are somehow associated with the animal, but not necessarily being ingested, because bacterial fatty acids can't be incorporated into animal phospholipids. And so there is a quick and dirty way of doing it. I mean, you could just do a neutral polar separation, but if you want to hunt that down, you can certainly do it.
Cool, that's good to know. We'll write down for when we get some money around.
Excellent.
Okay, are there any other questions? I think we're coming up on 4:30, so that was about what we had scheduled. But if there's any last questions. It doesn't seem like there are, so thank you. That was a really great talk. I really enjoyed it, and I don't know a lot about aquaculture, so that was really interesting for me. And we're really glad to have you for the seminar this week.
Thank you.
Okay.
All right.
Well, thank you,
everybody.
Thanks, Daria. Bye.
Thanks.
Bye.
See you.